Hey guys, hi. This is Sona Singh once again. Hi. So I know I have been a little late, and we promise we'll discuss a lot of stuff. So as of now, what we are trying to do is we would branch out to do three type of stuff. We'll start a series called Foundation. Foundation, the syllabus would be framed for students currently in their ninth and 10th. This will cover all boards, which includes CBSE, ICSE, Tamil Nadu, Maharashtra. So, irrespective of whatever board you belong to, we will cater to all the syllabus, and we'll start another series called NEET. Now, this will be for NEET students, that means currently in their 11th and 12th and preparing for NEET. Um, the materials would be there, but it will take some time to come for these videos to start getting published, which I'm expecting will happen by the end of February. And in the meanwhile, because you have your IOQJS and IOQB coming up, so we'll have practice sessions. So each practice session will have 4 to 5 problems or more if we could increase our efficiency. So if you have questions, you know already my Telegram, that is Sona Singh Biology. So this is on Instagram, this is on Telegram. You can reach me there. Now let's get started. So we are starting with the paper of 1718, and this is IOQB or slash NSEB paper. So let's get started without wasting any more time. So first question, it's pretty simple, not a little bit. So a veterinarian wanted to determine the pedigree of a puppy. Pedigree meaning the genetics, where did it inherit what from, right? So 4 male adult dogs had to be considered as possible fathers. So he wants to determine who is the father, right? And the result, how does it want to do that? By using DNA fingerprinting. Now, what is DNA fingerprinting? So, uh, this is your piece of DNA. You have double stranded DNA. You already know that. Now, DNA is, if you zoom out, and guys, don't worry, I'll be discussing the concept, not just telling you out the problem. So all is cool. Huh? So DNA is double stranded. What does double stranded mean? What does it mean when I say DNA is double stranded? Meaning it has 2 threads. So whenever you talk about a piece of DNA, it will have 2 threads. This each of these threads is going to be composed of small units joined end to end. So this one thread is deoxyribonucleic acid, and this deoxyribonucleic acid is going to be made up of deoxyribonucleotides, right? These are small units. Now, in between these small units, you have bonds, right? So these are phosphodiester bonds, right? So these bonds are called as phosphodiester bonds. Now, when you break these bonds, the deoxyribonucleotides go their separate ways. Now, how do we break these bonds? So as it appears, there's an enzyme, right? For everything, we'll have enzymes. So there's an enzyme called restriction enzyme. The reason we call them restriction enzymes is because these were discovered in bacteria. So there were bacteria which were, discovered by your infected. Sorry, the bacteria were very frequently infected by viruses, and these bacteria came up with a strategy to clip out to kill those viruses. So these were restriction enzymes, right? You have various of them, like you get have ECO-R1, that's one of them, or you get SAM, and there are many others, don't worry about them. So restriction enzymes are of two types. They are endo nucleases. Now this should make sense. Nuclease, it will break down a nucleic acid. Endo, it will break it down on the inside, meaning this is where they clip. This restriction endonucleases clip on the inside. So that means there's a second variety, exonucleases. Exo, on the outside, they will clip from the end, right? So we have these restriction endonucleases that can clip DNA from the between, from the middle. So because 99.9% of the DNA that you have is similar. So this is you and this is Amitabh Bachchan. You are going to have 99.9% DNA as same, right? Now, but there would be differences. The differences would be because you have different alleles. Remember that you have different alleles. And what are alleles? If you have forgotten, different alleles meaning different recipes, right? And that means because of slight differences, the restriction enzymes, when they try to digest the DNA, to break the DNA, they will digest the DNA. They digest the DNA in a very specific manner. What does it mean in a specific manner? That means they cut at specific points. They will not cut anywhere, right? They cut at specific points. Because there's only 99.9% similarity, meaning DNA of different people, right? DNA of different people will be dissected at different points. What does that mean? Let's take this DNA. So this is you and this is Amitabh Bachchan, right? So there are minute differences, and that is why the restriction enzyme cuts the DNA here in you, and the restriction enzyme cuts the DNA here in Amitabh Bachchan. So that means there are DNA fragments around this size, right? Those DNA fragments would look like that, and Amitabh Bachchan's DNA fragments would look like this, right? Now, what do you do with these DNA fragments? You can't see DNA, right? Okay, they are fragments of DNA, they are molecules. You can't see the molecules. So you make them run. How do you make them run? So how do you do all of this? So you take a cell, you first break a cell, you break the membranes, isolate the DNA. How do you do that? By using a specific set of solutions. You have various ways. One is by using phenol and chloroform, right? So you isolate the DNA and then you digest them using restriction enzyme. Now, this is once again your DNA and this is Amitabh Bachchan's DNA. Now, different DNA pieces will take different time to run across the gel. So this is a gel, and on this side you have the positive electrodes, and on this side you have the negative electrodes. The thing is, DNA is negatively charged. So all the pieces of DNA are attracted towards this end. But because of the different size, this big DNA will be slow. And the small pieces of DNA would be faster, right? So you get a print. So you get a print of differences in the DNA because of the differences in the sequence. This is one way of making or understanding the differences in the sequence. Okay. So in this whole method, what we were doing is we are understanding the differences in the DNA sequence, that 0.1% difference in the DNA sequence, by understanding or by visualizing the different pieces of DNA which were of different length because they were cut at different sections using the restriction enzymes. This is one of the way. In fact, this is the oldest way. Now the world has moved past it. We have different ways to do this and advanced ways to do this. But this technique was called as RFLP. Restriction fragment length polymorphisms. So by using the restriction enzymes, you create a print of DNA, right? So because you are made from the DNA of Papa and Mommy. So your restriction fragments, the fragments of DNA pieces that your DNA will have, must come from Papa and Mommy. That means all the fragments that you have must be there in either Papa or Mommy, right? So let's come back to our question. The question is, Lily is the mother, right? And Sweetie is the puppy that you want to understand that the pedigree of. So all the restriction fragments in Sweetie must come either from the mother or from the father, right? So, okay, Sweetie has this, and that's found here, and that's found in Jim, Jack, and Jill. Okay, not a point of difference since this is already there in Lily. This fragment is already there in Lily. Not a point of difference, not important for us. These are important for us. Why? See, why I'm saying that that's not important, because it's already present in mother. So if it's found in father or not is not really helping us, right? Now, we are interested in fragments that's found in the puppy but not found in mother, because in that way they must come from the fathers. So this is an important one. And what else? This is an important one. And this is an important one, because these fragments are must come from fathers and not from mothers, right? So we can see Jim and Jam make all round perfect fits, right? Here we see Jam is a better candidate. Once again, we see Jam is a better candidate. And once again, we see Jam is a perfect candidate, right? So fight is between Jim and Jam. Jim and Jam, but while in this position, let me mark the positions 1, 2, 3, and 4. So second, third, and fourth means that you can have more fragments, right? So we confirm Jam is the father because these fragments are there from Jam. So the correct answer for this question would be Jam. Guys, a quick word. Please ensure that you like, share, and subscribe. Very importantly, subscribe to the channel, because the length of the videos that I can put up will depend on the subscriptions. I think before I have 100 subscriptions, I can only put up 15-minute videos. And I think after 100 subscribers, there's YouTube policy and whatever. So please ensure that you subscribe to the channel. I'll try to make the videos as long as possible and eventually turn them into 1 hour lectures, but I need subscriptions, guys. Okay. Also, one important thing, choice of language. So right now I'm using only English, right? Now I am going to give you 3 options, which you must reply in the comment section: English, Hindi, or Hinglish. So what do you prefer? Do you want me to pull separate videos in English and Hindi, or do you want a mix of English, or what, right? Okay, so that's the first video, and I think I am out of the time, so I want to do more, but I think I can only upload 15-minute videos. So I'm stopping here and we'll continue in the next sessions. Thank you so much. Don't forget to subscribe.